I can start, yes? Share screen.
Wait a second. The prorector will first open the ceremony.
Okay.
Welcome all to this ceremony. I hereby open this academic ceremony in which Sebastian Sanduleanu will defend the academic thesis Radiomics Imaging Biomarkers from the Perspective of Tumor Biology. I'd like to welcome everybody, the members of the supervising team represented by Professor Lambert, the members of the Corona, which I will introduce later to you, and of course the candidates, and the audience that is following this session by live stream. First of all, I would like to give the floor to the candidate. May I invite you to present a summary of your study and the conclusions of your thesis. The floor is yours.
Okay. Can everyone see it?
No, not yet.
And then I have to...
The share screen in Zoom.
Zoom support, and then screen, and then share screen now.
Is this right
Yes.
for you? Go ahead.
Yes, thank you very much. Dear Pro-Rector, dear members of the Corona, dear family, friends, and audience. In the coming 15 minutes, I will shortly introduce my thesis titled Radiomics Imaging Biomarkers from the Perspective of Tumor Biology. Or in Dutch, interpretatie van tumorbiologie met behulp van radiomics, beeldvormingsbiomarkers. My thesis can be broadly subdivided into 6 distinct parts, depicted here. It consists of an introduction, 3 main topics, and a discussion and final summary and impact chapter. In light of the brief time, we are only going to discuss the results from the 3rd part of this thesis book. Oh, I don't know what happened here. Why is it, sorry. Yeah. Apologies. Okay. Let us have a closer look at the tumors that were investigated in this thesis. Broadly speaking, 85% of lung cancers are histologically of the non-small cell type, while 15% are of the small cell type, depicted in this pie chart in red. Our main focus in this thesis was on the more prevalent non-small cell type of tumors. Lung cancer was in 2020 in the Netherlands diagnosed in 6,400 women and 7,500 men. And in the same year, there were these many deaths, making it the most lethal cancer in the Netherlands upon diagnosis. The incidence of head and neck cancer is lower than lung cancer in the Netherlands, with a little over 3,000 new cases in 2018. And this type of cancer is found primarily in the oral cavity, larynx, and oropharynx, circled in the left images in red. In the right image, one human figure depicts 10 patients, and the patients in the red box depict the top 3 sites where the cancer is found, namely again in the oral cavity, larynx, and oropharynx. The mortality of head and neck cancer is low, but has been increasing in both men and women since the 1990s. I would like to now define 3 important terms in my research of these cancers. The first term, tumor hypoxia, is defined as low oxygen in tumors. To be more specific, tumor hypoxia, again defined as low oxygen within the tumors, is caused by abnormally formed and occluded blood vessels, leading to unstable blood flow and inadequate nutrient and oxygen supply, as can be seen in the blue regions in the right figure. Hypoxia is to some extent present in all tumors and typically is untreated by conventional therapies such as systemic therapy and radiation therapy, and it promotes mutations, tumor invasiveness, and metastasis, leading eventually to treatment failure and poor prognosis. Research on ways to measure hypoxia started nearly 100 years ago with the microscopic examination of cancer cells and their surrounding blood vessels. Hypoxia research has been from the very beginning closely related to radiation therapy in cancer. In the 1930s, the first radiation treatment started at hospitals all over the world. At the same time, the first microscopic studies hypothesized that cells at the margin of the tumors that receive more oxygen supply are more sensitive to radiation than oxygen-poor or dead, also called necrotic cells, in a tumor. In the decades after, radiobiology studies measured the tumor oxygen content through probes, and therapies with oxygen or oxygen mimickers were studied in different cancers, often with promising survival results. Nevertheless, as the probe only measures the oxygen content of a small part of the tumor, you can imagine that this does not confer information about the oxygen content of the entire tumor. In the 1980s, a special type of scanning technique called PET scanning was introduced, making it possible to visualize hypoxia in 3D. Injecting a mildly radioactive substance into the vein of the patient that specifically binds to low oxygen regions and lights up on the scanner was from this moment possible. Several of these substances were developed, which were reliable in detecting hypoxia, but were highly expensive for the patient and time-consuming for the hospital technicians. These substances therefore never entered clinical practice. In this thesis, we obtained a method to measure hypoxia based on these scans. Later, several hypoxia staining markers, such as carbonic anhydrase 9 or CA9, were studied, and in the early 2000s, the first DNA studies of hypoxia were performed. Unfortunately, they were shown to not accurately measure oxygen content in the tumor, as these markers have tumor-specific activities and are expressed with a time delay after onset of hypoxia. So to summarize, after almost 100 years of hypoxia research, we still have an unmet need to accurately measure this entity in the clinical setting to be able to adapt our therapies accordingly. To come to the second term, big data in radiology alludes to the use of automation in studies of large amounts of medical images. With an ever-increasing amount of medical images from cancer patients, also known as big data, such as the ones showed in the previous slides, scientists in the past decade have found the need to use more sophisticated computer models to solve the scientific tasks at hand. Artificial intelligence is a branch of computer science dealing with the simulation of intelligent behavior in computers. Machine learning is a subset of artificial intelligence that provides the ability to learn automatically to improve from experience, whereby an intermediary human is not per se needed to teach the algorithm. Think as an analogy of a child that can learn the types of fruit from labels in a picture book, or on his own by simply observing the colors and patterns. The former kind of machine learning models were primarily used in this thesis. Lastly, there is also deep learning, which is a subset of machine learning which uses so-called neural network models to analyze different factors with a structure that is similar to the human neuronal system. To come to the third important term, radiomics. Radiomics is related to measurable medical image descriptors. So what is radiomics? Radiomics is a quantitative imaging approach that extracts a large number of features or quantitative imaging descriptors from medical images. It comes in two types: handcrafted features based on mathematical descriptors of the tumor shape, intensity, and texture, and deep learning features, which are analyzed with neural network models. As previously mentioned, the most reliable yet time-consuming and costly method to measure hypoxia is with a group of injectable, mildly radioactive substances, which then can be visualized on a scanner. Imagine a patient with cancer that requires therapy receives a routine scan prior to treatment, which consists of a computed tomography scan to locate the tumor anatomically. Furthermore, most patients also receive a PET scan after injection of a mildly radioactive sugar-bound substance called FDG. which enters the sugar-hungry tumor and emits energy packets, which then can light up on a scanner. After a few days, according to this information, the radiotherapist and oncologist can select the correct treatment. A few days after this routine clinical measurement, the patient could receive another injection with a substance that now binds the oxygen-poor regions of the tumor, and only in these conditions emits energy packets to the scanner. This is called hypoxia PET. This is at the moment not the standard procedure in hospitals, as it is expensive and time-consuming. Once patients are found to be hypoxic, their standard treatment can be modified by providing them with medication that selectively targets hypoxia. New generation of these drugs are currently under investigation in human clinical trials. In one study, we wondered whether hypoxia is measurable with radiomics. Our hypothesis was that radiomics features from routine computed tomography and FDG PET scans are able to accurately detect hypoxia, waiving the need for expensive and time-consuming hypoxia PET. So as an example, we wondered whether this lung tumor on a CT scan is hypoxic or not without performing the hypoxia PET scan. Novelty of this study was that tumors were defined as being hypoxic according to a high hypoxia PET tracer uptake of more than 20% of the total tumor volume. So the red region here should be more than 20% of the total tumor volume to speak of hypoxia. The main results were that reliable and robust to change imaging features for hypoxia were obtained with external validation dataset areas under the curve, which is a metric for the model performance, in some cases of above 0.8. The threshold for clinical relevance is also mainly cited to be above 0.8. Furthermore, interesting features were found on computed tomography linked to highly different gray values within the tumor and thus a high heterogeneity of the tumor. In the PET model, high percentile total lesion glycolysis features were found. So what does this mean? As expected, tumor cells switch metabolically to the high glucose turnover to lactate, even in oxygen-poor environments. This is depicted in the image on the right, and is also known as the Warburg effect. And these features were therefore found in our models. General conclusions were that combining biomarkers from multiple sources such as DNA markers and imaging markers constitute a cost-effective and reliable method for hypoxia measurement. Benchmarking radiomics steps were also successfully implemented in this study, such as combat feature harmonization. In another study, we looked into early treatment hypoxia changes in head and neck cancer in a head and neck cancer population. The study workflow from this head and neck cancer population from two Dutch trials was as following. Prior to radiotherapy, a planning computed tomography was performed to accurately deliver the radiation to the tumor in the patients. Subsequently, prior to treatment, a hypoxia PET scan was acquired. Patients then received treatment with radiotherapy, chemoradiation, or immunoradiation up until week 2. Afterwards, another hypoxia PET was acquired, and patients continued their treatment. We wanted to see whether there was an increase or decrease in hypoxia in the first 2 weeks of treatment, and whether there was an association in the hypoxia PET signal between prior treatment and the week 2 scans. The results were that there were large differences in PET signal found between the first and week 2 hypoxia scan, signifying the rapid change in hypoxia early in treatment. No decrease in hypoxia in the early stages of treatment identified patients with a poor prognosis, or with other words, Patients with an increase or no change in hypoxia lived shorter. This brings me to the conclusion of this thesis and future directions. The United States is taking the lead. As of this month, there are approximately 146 artificial intelligence algorithms approved in the United States, and some of them are already on the market. Explainable and generalizable artificial intelligence solutions are required, as well as collaboration between medical specialists and hospital technical staff. In these studies, machine learning applications are unlikely to replace many human skills, such as complex reasoning, abstract problem solving, judgment, empathy, and counseling. So we should focus on human-centered solutions with a strong ethical and legal underpinning. Thank you for your attention. I give now the word back to the prorector.
Thank you very much, Mr. Sanduleanu, for this presentation. We will now start with the opposition, and the opposition will be opened by Professor Motagui, who was the chair of the assessment committee of your thesis. He's Professor Nuclear Medicine at Maastricht University Medical Center. Professor Motagui.
Thank you very much, Professor Martin van Baak. First of all, I would like to congratulate you, the candidate, and also the promotion team on this excellent compilation of these scientific work into the direction of validation of radiomics approaches. I think it's impressive. There are 10 papers, and there are very different aspects you have touched in your papers. Nevertheless, of course, we are not here to only congratulate you, but also to discuss a little bit and to see into what direction you can evaluate also your own results. I would like to start with a rather easy question probably for you, but on the other side, a question that is also a lot of people are asking. So if you would have the choice to really implement radiomics into the clinics, what kind of approach would you do? Which modality would be your first choice that you would like to have to really go into to bring this into the clinics? Because as far as we have it right now, there's a lot of discussion on radiomics, but only very few aspects are already in the clinics. So what would be your approach to bring it to the clinics, and which modality would you promote?
Highly esteemed opponent, thank you for your kind words and for your question. To promote radiomic studies in clinic, I think we should go by an approach in which we first have the implemented in a clinical setting with the lowest risk of any adverse events or effects for the patient. It could be, for instance, that with these radiomic studies, you could predict a certain treatment outcome in clinical trials, such as with my hypoxia signature. You could predict whether a patient responds or not to drugs that target hypoxia. So this would be a first step, I believe, the stratification of patients in certain trials with certain drugs that are tolerable and safe for the whole patient population, just to see which patients have the highest effect, and whether we can stratify that based on the hypoxia or on the radiomic signature in general.
Okay, so that's certainly one aspect. The other aspect, since you have used CT, PET, I don't think you also used MRI in your studies, but anyway, so if you have these modalities available next to, of course, the biological parameters you also included in your analysis, what would be then the safest approach to use? So which modality would you like to have in a first step? What would your idea be on that?
Well, highly esteemed opponent, again, thank you for the question. I believe already tumor volume, for instance, on a CT scan is a highly explainable and well-documented metric that could definitely be used to on a CT scan, that could definitely be used to follow up patients during their treatment and see whether the volume actually changes of the tumor instead of just the longest diameter. Because that is not always the RECIST 1.1 criteria are not always the very sensitive to the changes, the volumetric changes of the tumor, which I think are more important or more interesting to study. So I believe that would be a first step.
Okay, so your answer is into the direction, so you would rather go for the CT, right? So if
I believe, yes.
Try to translate your
I believe, yeah.
Okay. Great. So then on the other side, you also have these different kind of biological parameters. Of course, you have the PET different traces. I would not like to talk now about this, but rather on the biological parameters. So, because in your title, you write, the title of the thesis is from the perspective of tumor biology. So, of course, hypoxia is one aspect, but there are a lot of other aspects in a tumor that could be of importance for these kind of approaches. So, what would you think would have been maybe an additional add-on to your analysis in the approaches you had so far?
Well, I believe that we also looked into other biologic entities, especially in chapter 10, with the peritumoral radiomics. We looked at changes around the tumor border in the peritumoral region, which may be linked to malignancy-associated changes that could help spread the cancer further and metastasize faster. So we've also looked into other biologic entities. But of course, it is very difficult because you need, you need a ground truth measurement of biologic substrate. So in this case, you could co-register the images with histology images and see that it actually detects something that has a biologic meaning, which I was not able to do with this study. In this study, we only looked into the prediction of outcomes, local regional recurrence, distant metastasis, overall survival. But I believe those are very multifactorial and highly difficult to just detect only based on a image.
Yeah. That's the point that you have to deal with if you're going to radiomics. I think my time is over. I thank you very much for your. I would like to give the word back to the prorector.
I have to unmute myself. Thank you very much, Professor Motagui. The opposition will be now continued by Dr. Lobbes. Dr. Lobbes is from Zuiderland Medical Center and Maastricht University Medical Center, and he was also a member of the assessment committee. Dr. Lobbes.
Thank you, Madam Pro-Rector, for your introduction, and I also want to extend my congratulations to the PhD candidate for really writing a nice overview of very novel strategies, which I, as a clinical radiologist, are all too familiar with, not primarily with the technical insights of it, but I see the useful applications already at the horizon for many different indications in medical imaging. And the studies that you did in your thesis are only a very small part of applications that are potentially possible. You can think in diagnosis, workflow applications, quality criteria. So the sky's the limit in this field of expertise. With that, I also would like to extend the same congratulations to your promotion team. I know them as a very adequate group, and I really enjoy working with them as well, joining their technical expertise with my clinical expertise, which is always very, inspiring to do. But aside from all the compliments, I also want to be very critical, as I'm supposed to be, and I want to focus with you on chapter 9 on the preoperative CT-based radiomics combined with intraoperative frozen section. That will be my main focus of attention. I have several critical comments or questions regarding that study, and perhaps you can give me more detail or sort of take away my anxiety. First of all, why did you choose to use frozen sections and not biopsy? Both have their limitations, but for me as a clinical radiologist, I would think biopsy is more easy to get in an outpatient clinic instead of already doing a surgery. So what's the reason behind that?
Esteemed opponent, thank you very much for your kind words and also for your question. With regard to the frozen section results instead of biopsy, this is a study where patients were selected from Chinese medical centers, and there is this. They had this data, and it was common practice to get these results from the frozen section. It would be possible to do a biopsy, but I believe it's not in all cases feasible. So in light of having this data with this large amount of patients with, with actual resection specimens, yes, this is what we were able to do.
So this is what you had to work with.
Yes, exactly. Yes.
So and when I looked critically at your methods, you have a very fixed set of inclusion and exclusion criteria. And when I look at figure 1 of that study, which is sort of the patient flow chart, when I do a quick calculation, I see that you, based on these criteria, you have a dropout rate of the initial patients of 55%. So more than half of the patients you initially thought was interesting end up in the final analysis. Does this lead to any selection bias, and how would it affect the results, do you think?
Yes. Highly esteemed opponent, thank you very much for your question, or esteemed opponent, sorry. So the inclusion criteria for this study were that primary lung adenocarcinoma confirmed by pathology, and also pulmonary nodules without a visible cavity. So I believe there were a lot of patients with a visible cavity in the pulmonary nodule, which then could influence the radiomic features a lot and not give us enough information. So I believe that's why a lot of patients were excluded. And probably also because patients had a prior history of chemotherapy, radiation therapy. The time interval between the CT examination and surgery was more than 2 weeks. So yes, there is definitely a bias here. But how that reflects in the model that we trained, I don't know, because we did not have this clinical information.
If you talk about radiomics analysis in those pulmonary nodules with or without cavities, you say, well, we don't have any solid tissue to work on and to have anything, but that's how it works in practice. And sometimes you do not know it beforehand on CT whether there will be cavities or not. Do you think if I would include these patients as well, your radiomics model would become useless, or would you still think there's any value for it?
Mm-hmm. In my personal opinion, it is important to have a general broad vision and to include everything from a real life situation, how it would happen in a real life clinical setting. So I believe that would definitely be worthwhile to have a look at. Yes.
Okay, and with the permission of the pro-rector, one small question. I think there's 1 or 2 minutes. I read that you had the delineations being done by one expert chest radiologist, and I see in the office, I probably know who it was, which is a very accurate radiologist in my opinion. But you also had a subset being drawn by, I believe, a second radiologist and then somebody expertised in delineations. But I don't see that coming back again elsewhere in the manuscript. So my question is, where's that data, and how? Perhaps you forgot, or for a word count you were able to include it. But how would you present it? With what kind of analysis would you present that data? And did you have that data? And
yes, so we had this data from a second radiologist, and we presented this data by excluding features which from the radiomics model, which had a low intra-class correlation based on the segmentations. So we excluded those features just to make sure that there is more, the segmentation is more accurate, and that we were able to better account for segmentation differences.
Okay. Well, I think seeing the time, I thank you for your questions. I'm very happy with them, and I would like to return the word to the program. Thank you.
Thank you, Dr. Lobbes. The opposition will be continued by Dr. Walsh. Dr. Walsh is from Radiomics in Liège, Belgium. Dr. Walsh.
Thank you, Prorector Professor Van Back. My congratulations to the PhD candidate and to the supervising team. You've prepared a fine work and body of evidence for radiomics, and it was really quite stimulating for me to review the work from conception through to the final papers which have been produced. And that's where I'd like to begin my opposition, is on the future perspectives. You prepared a fine paragraph at the end of your thesis, which touched upon the challenges being faced by people working in this area of research, how they'll cross the translational gap from great ideas to real clinical impact. One of the key points you make is, you said there's a lack of harmonization, and throughout your thesis you pointed at 2 ways to address this in the clinic: harmonization with respect to images and practice. One was on methods such as combat, so on the machine learning side to sort of correct for the data which we're dealing with. And the other is on the human side, where we change the processes, that we get people to collect and acquire images in the same way. Could you please outline for us the roadmap, if you were given the authority and power? How would you get a complex multi-stakeholder environment, such as a hospital, but a healthcare system nationally and internationally, to rally around a harmonized approach? How would you lay that out for us? Could you please take us through your thinking about why we could get to standardized image protocols, or if we don't need them, what can we do on the back end with the machine learning?
Esteemed opponent, this is a very interesting question, and it is very much an active phase of research at this moment. The harmonization of not only the radiomic imaging features, but also harmonization initiatives of the images themselves, as they are acquired by different scanners. And I would like to reference an article by Mali et al., Journal of Personal Medicine 2021, in which different harmonization methods, both in the image domain as well as in the feature domain, are described, and where we are currently standing on this topic. So my first observation is that that it is also possible in the image domain, for instance, by using the raw scanner data, or by using the filtered back projected images and then applying certain deep learning algorithms to them, such as generative adversarial networks. We are able to harmonize them, to harmonize these parameters very well. But this has not, first of all, not been already implemented on large scale in radiomics studies. And second of all, the individual vendors of these different scanners have not picked up on this yet. I would believe there is no awareness yet in all these efforts. So I believe that is the first step to improve our radiomics workflow with these methods, and to secondarily create awareness that these methods are possible and that they could lead to implementation.
Okay. Thank you very much for that. I'd like to just quickly tease that answer out a bit further, because if we deal with the raw data, as I mentioned, there are multiple stakeholders. So the raw data is coming from the vendors. That isn't accessible to the clinical staff usually, typically. It can be accessed, but it's restrained and restricted. If you solve the situation there, it's on the imaging vendor side that the solution is. So how would that translate to working with a therapy provider, such as someone who's developed a hypoxia-activated prodrug? Do you see how there could be a gap there, again, between, let's say, a vendor wanting to implement this solution for the benefit of a therapy provider?
I believe that this I'm not aware of such initiatives, but I believe there are very good initiatives if that is possible indeed.
Okay, so partnership in an ideal world, we would all be on the same team, and these companies would work together for the benefits of the patients and the clinicians.
Ideally, because they are all standing to benefit from that.
Yeah, indeed, improving. And to go beyond that, then on the human side, if we forgot about processing the data, how can we get people to adopt these solutions? So just something as simple as people adhering to protocols to get better CT images or to get better. How would we do that?
There are several initiatives in doing that. For instance, the Earl PET initiative to standardize PET images according to several protocols. So this is already being done at this moment in larger and larger amounts of centers worldwide. But I believe we're not there yet. I mean, there are many. Even if you have these different protocols and they're required within a certain set of parameters, of course, there are these different protocols which also impact the imaging features that we're calculating and of course, the outcomes of our models. So there is still a large gain there.
Okay, absolutely. And if I can give you a dichotomy, so ideally we get to the solution, but there's two ways I can put forward to you. One is through education, that we do a persuasive act, that we convince the radiologists, the oncologists, all of the people involved in care that it really does pay value if we all work and apply the same image acquisition, because then we can compare like for like and we get to a better place. That's one way to go. The other is to regulate it or mandate it, whereby an authority comes in and says, well, actually, we're restricting your freedom to choose. You have to image in this way for the good of the entire healthcare system. Which of those approaches would you advocate for? The regulatory constraint side or the persuasive side?
I would always go. I believe you should always go for the persuasive side and get people on board based on educating them and based on showing them the benefit of such practices. I don't see, and I'm not a legal expert, but I don't see ways in which you could enforce this either in all the countries. So that's a very legal, from a legal point of view, a very tricky question, I guess.
Yeah, indeed, the regulatory space is a highly intricate and sophisticated area, so no problem not understanding the legalities. Thank you very much for answering the questions. Appreciate your ability to stretch beyond the normal scientific zone to deal with the practical issues. So with that, my thanks, and I give the word back to the prorector. Thank you.
Thank you, Dr. Walsh. The opposition will continue by Professor Nicolai. Professor Nicolai is from the Universitätsklinikum Freiburg in Freiburg, Germany. Professor Nicolai.
Thank you, Prorector. First, let me also congratulate the candidate for his remarkable thesis, which I enjoyed reading. We had the candidate over for a departmental talk 3 years ago in Freiburg, and he presented us a very nice presentation on quality assured radiomics. And it's very nice now to be present also in the final stages of his research and in his thesis. And I want to congratulate on achieving such a very nice thesis. And obviously, I also want to congratulate the committee and the supervisors on this thesis as well. Being a radiation oncologist, I was very intrigued by the chapters that you wrote on hypoxia. And as you also demonstrated or presented in your initial presentation, hypoxia is one of the key problems in radiation therapy, and it's one of the key factors for resistance to radiation therapy. So in this respect, I was wondering how your radiomics approaches may help to devise strategies to overcome this resistance. So far, the quality of the features may be very good, and you have done a lot of quality assurance, but by deducting radiomics features from the actual images, you also remove some spatial information that may very well help us as radiation oncologists to define high-risk regions that we may use to increase treatment, to escalate treatment, or on the other hand, to de-escalate treatment. You have discussed some aspects, for example, adding hypoxia-activated prodrugs to the solution and to combine systemic treatments. I was wondering whether your analysis will also provide some way of improving focal treatments or to stratify patients for certain focal treatment approaches.
Highly esteemed opponent, thank you very much for your question and for your kind words. With regard to focal treatment, radiotherapy, dose escalation studies, for instance, you could, based on this radiomics signature, definitely find patients that have a high hypoxic fraction or hypoxic volume. And by finding these patients, you could, with a really high hypoxic fraction, you could design dose escalation studies and specifically select those patients which will benefit from, for instance, dose escalation or from hypoxia-activated prodrugs or from other hypoxia-modifying treatments. I would like to cite a study by Kutte et al. In Radiation Oncology 2021, in which they actually simulated with proton therapy in non-small cell lung cancer. They simulated hypoxia-targeted dose escalation that can mitigate the tumor control, so mitigate TCP reduction due to hypoxia-induced radioresistance, while simultaneously reducing the normal tissue complication levels. So there are already these simulations have already been done, for instance, with proton therapy. The radiomics hypoxia signature could also definitely find patients that, for instance, that have a high level of hypoxia. Then you could do a hypoxia PET-CT, and based on that, change your treatment, your dose escalation, for instance.
Okay, thank you. Let me be a bit more provocative. How would you technically do this dose escalation? Would you just increase the dose to the whole tumor, or would you somehow have a way of selecting high-risk areas within the tumor that may benefit? Because obviously, if you escalate the dose too much to the tumor, you will increase very much also the toxicities to the surrounding normal tissues, which is a key problem in head and neck radiotherapy.
Thank you very much for the question.
Maybe you can try to give a short answer.
Oh, short answer, yes. Really short answer, sorry for, short answer would be that you could do it two ways. You could, for instance, target the hypoxic regions. If those are close to healthy normal tissues, you could target those with hypoxic activated prodrugs, and then give a higher dose, for instance, to the non-hypoxic regions. There are several ways to go about that. It's still in the realm of possibilities and has not been, from what I know, has not been attempted in a large set of patients in radiation oncology, and we don't know what the effects are on toxicity and on tumor control probability on large sets of patients. So it is a very interesting topic of research, but I don't. We still have to see what the possibilities are at this moment.
Okay, thank you very much. Regarding the lineup of the other opponents and the advanced time, I would pass back to the prorector. Thank you.
Thank you very much, Professor Nicolau. The opposition will be continued by Professor Zips from the University of Tübingen in Germany. Professor Zips. You have to unmute yourself. Yeah.
Thank you, dear prorector. I have to echo the congratulations to the candidate and the supervising team. Very timely topic, very exciting technology and unmet clinical need, as you have nicely explained. I would like to discuss, as a radiation oncologist treating patients with head and neck cancer, I would like to discuss chapter 10 with you. And there you have shown in your data that predictive models, including clinical factors and also radiomics, may help to inform treatment decisions for head and neck cancer patients. My question is, could you explain us by the numbers, maybe in the tables 2 and the others, what the added value of the radiomics, in addition to the established clinical models for prognosis in these patients? How can we derive this from these numbers? The added value of radiomics.
Highly esteemed opponent, thank you very much for your question and kind words. As can be seen in table 2 and 3, what we present here are the C-index or the performance metrics for the different models, be it the prognostic clinical models, the treatment-related clinical models, so that would mean the chemotherapy regimen, the completion or not of chemotherapy. So those were all variables that we used, and also the peritumoral radiomics performances. And as you can see here, the radiomics performances were all very good in the training data sets, but performed very poorly in the validation data set. Now, the question is, why is that? And first of all, I have to say these findings are in line with other studies, such as Du and al. From non-small cell lung cancer patients. This is a study that also looked into peritumoral radiomics and was also presented with negative findings in +1. So there they also didn't find any high benefit of peritumoral radiomics. The question is, why is that? So these malignancy-associated changes on the tumor board, on the peritumoral region, are mostly changes in the nucleus of the cell, and very subtle changes, which I believe we cannot really well pick up on radiomics, with radiomics, which are higher voxels and don't potentially confer the changes that happen on a subcellular level. So I believe that is my answer.
Okay, thank you very much.
The opposition, thank you, Professor Zips. The opposition will be continued by Professor De Ruyscher from Maastricht Clinic and Maastricht University here.
Thank you very much. So first of all, I want to congratulate also you, the whole team, obviously, but also your family, because I know that PhD is also quite heavy burden for the family of the PhD student. So I want to congratulate them as well. But of course, I will ask you some questions, and I will start with chapter 7, page 165. So I'll give you some time.
I am there.
Yes, okay. And that's about point 2.3, the image evaluation on HX4 scans. Now, my question is actually about the rigid registration. So you mentioned that you have 2 time points and that there's a rigid registration using the Mirada software. But of course, if you have rigid registration, because of the volume changes, there will be some differences, of course, between 2 time points. How did you deal with those changes? Because it's a rigid registration.
Highly esteemed opponent, thank you very much for your questions and for your kind words. We dealt with rigid registration by also visually looking at the tumor on the CT scan and trying to adapt that volume based on those changes. Definitely Mirada software has also a really good deformable registration method. But we used from literature and from our at that moment understanding, we used the rigid registration approach, which I believe, yes, there are some problems there with the
Exactly.
matching of the volumes. Yes.
Exactly, because if you think about later applications, wouldn't it be good to have the best available imaging at the different time points? Because in general, what we do in radiation oncology, that is, we start with an optimal imaging at the time of the CT planning with registration with MRI scans, PET scans, and whatever. And then we take some CT scans without contrast somewhere during the course of the treatment. And of course, how would you deal with that problem? How will you really make sure that what you delineate is remaining tumor, including all the artifacts which are induced by radiation as well?
Highly esteemed opponent. I believe that, and I don't know, I don't have exactly a reference in mind, but I believe also the way radiation oncologists sometimes delineate the gross tumor volume. Sometimes they overestimate certain regions and underestimate other regions. So I believe the best way at this moment would be to have multiple experts delineate these or segment these regions. And then see how the rigid approach works, the deformable approach works on, let's say, the contrast enhanced CT versus the non-contrast enhanced CT with poor scanning parameters. And that is, to my knowledge, has not been studied yet, but it's my humble.
Okay, thank you very much. We could discuss this much more in depth, I think, also based on some own studies. But I will move to page 169 and on table 2 and figure 1 and so on. So I have some question about those results also in chapter 7. So I see there, if I read it well at least, that is that at baseline, the T stage, the location of the tumor, because it was a heterogeneous group of patients, of course, the hypoxic fraction at baseline was actually not related to local progression-free survival, neither to overall survival. Why would you think that is the case? I would think that if you have a bigger tumor, if you have a bigger hypoxic fraction, that it would relate to well, a worse prognosis. So how would you explain these results? Or do I read it wrong? It's also
I believe there are several things to be said about it. First of all, these are small data sets, so it's only you're training a Cox model on a very small set of patients, only 33 on baseline and even less on 28 in the week 2. That's probably one reason. Secondarily, you see there are some some p-values that are significant prior to adjusting for multiple testing, such as WHO performance. But that is after adjusting for multiple testing. That's it's not significant. So that's an interesting question. And for instance, what we know from the study from Zips et al. From 2012 is that they did found significant baseline parameters and week 2 parameters, but there was no adjustment at that moment for multiple testing.
Okay, thank you. And my second question is, what is the relation between the hypoxic volume and the GTV of the primary tumor?
The hypoxic volume
At the same time points. So did you calculate the correlation coefficients?
Between the GTV and no, I did not calculate that. No.
Yeah,
no. Because there are some studies, for instance, by Zegers et al. 2014 clinical cancer research that show that hypoxic tumor volumes are smaller than metabolically active volumes or GTV volumes.
that is correct, of course, I know that. But on the other hand, it may well be that, as an hypothesis, that a bigger tumor has also a bigger hypoxic volume, and hence it may also be that decreasing the hypoxic volume due to the radiation or the chemoradiation, whatever, during the course of the treatment, you have smaller volumes, and then what you are looking at is actually your hypoxic volume maybe becomes smaller, but your GTV also becomes smaller, and it doesn't add anything to the prognostic value of just looking at the GTV. What is your opinion about that? So you are doing a lot of difficult work where you could just look at the volume of the tumor.
That's an interesting question. Of course, that's one of the major 4 R's in radiotherapy is the reoxygenation. So when the tumor shrinks, because of the fact that the volume shrinks, you have more oxygen going to previously hypoxic regions, and that can definitely improve the outcome. So that's what fractionation is based on. And but we did not do this analysis.
Okay, thank you very much. Because of the time, I give the word back to the co-rector. Thank you very much.
Thank you very much, Professor Ruijser. Then we go to the last opponent of this session, which is Dr. Le Villain. He's from the Free University in Brussels, Belgium.
Thank you very much, Madam Proctor. Dear candidates, I will also agree with the rest of the opponents and congratulate you and the supervising team for this very interesting work. My first question concerns the different clinical trials that you perform in your PhD thesis. And regarding extraction of radiomics feature, we see that there is lots of preliminary steps before the extraction, such as image acquisition and standardization, image resampling, changing the number of bin or the size of the bin, and there is lots of technical aspects into this extraction. In these different studies, how did you settle these preliminary steps? Did you try to harmonize that? Did you try to standardize the whole process in your different studies? Did you try to fine-tune and see how changing one part of the methodology could impair the different signatures?
Mm-hmm. Hi, esteemed opponent, thank you very much for your question and your kind words. For instance, in chapter 5, and I'll have to go to the exact page. First of all, that would be on page 104 and 105. You can see the different the CT image acquisition parameters and how they relate to all the data sets that were used in the training and validation of these models. Yes, there are several steps. There is the resampling of the voxels. There is the problem with the feature harmonization, and for that we used combat feature harmonization method, which has been validated on phantom studies and has been shown to work really well. And then there were, let me see where we can find that.
Please unmute yourself, Dr. Le Villain.
Sorry, I click next to the button. Yeah, did you try to use the same methods across the different studies?
Yes. For instance, in chapter 12, we also used combat feature harmonization, also the same resampling method, and also the feature selection methods for the selected features were also quite the same in most studies. For instance, we looked at training out-of-bag cases with the highest accuracy. So that amount of features were selected. So we're trying to be consistent as much as possible throughout the studies.
Certainly you adapt between PET studies and CT studies.
Obviously, yes.
Okay, I don't know if I have time yet because I see that the time is running to ask an additional question. Maybe I will go to the short one. As mentioned, your PhD thesis title is Understanding Tumor Biology Through Radiomics Features. You detailed in the introduction that there is N-craft radiomics features, a deep learning radiomics feature. How do you see the future of deep learning radiomics feature to understand tumor biology?
There are several deep feature studies, deep learning studies out there, also for tumor hypoxia. That's based on multiparametric MRI, for instance. And this is a study by, I have to check, the MRI study, I don't have that here, but there are also for hypoxia, there is also a deep feature a method.
Do you think that these deep learning features are suited for understanding biology, predicting biology, and making hypothesis-driven studies?
There is you apply certain filters with deep learning, and that can give you some more information about a certain underlying tumor biology. Yeah. So I believe yes.
Well, Mr. Sanduleanu, as you can hear, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request you and the other viewers to await the results of our deliberation and our return. I think we are all back. Mr. Candidate, Sebastian Sanduleanu, the degree committee here present online has assessed the quality of your thesis and of your defense. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Lambert is authorized to confer upon you this academic distinction in accordance with Dutch university custom. I invite your supervisor now to take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I do.
By the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Sebastian Sanduleanu, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university shown by the beadle. So, Sebastian, congratulations for your diploma. As you know, it's the highest degree that a university can give. And I remember very well when you started with us, I think it was in Maastricht clinic in that time. And then initially it was planned that you would do a more sort of a clinical project. And then finally, for all sort of reasons, you ended by doing quite a lot of machine learning, statistical analysis, etc., probably more than you expected when you solicited for the job. And then I'm actually very impressed by the progress you made. You learn a huge amount. And then, you know, and so the member of the committee pointed out that you had quite a difficult task because the topic of the thesis was extremely broad. And we've seen that today. You have had some question on radiotherapy while you are following a training of radiology. You had a question on radiobiology, question on machine learning, etc. And you managed to give a decent answer to sometimes very good answers to all those questions. So you are now following a training of radiology in Zuiderland, and we are very happy with that. We are very happy then when our PhD students continue their life. And here we hope that by training you, we will participate to the implementation of AI in the clinic, because I believe that I use also the word I believe, like you did doing your defense. Now, I think that we need people like you to implement artificial intelligence in the clinic. And so I wish you a lot of success and lots of happiness.
Thank you very much. Thank you very much. Yes.
Also, on behalf of the Board of Deans of this university, I congratulate you, Dr. Sanduleanu, with the honor you have acquired. And I also like to congratulate your friends, family, colleagues with this achievement. I'd like to thank all the members of the Corona for their contribution to this session. It's a pity it had to be online, but I hope you still have good memories.
It's indeed unfortunate, but it was a nice event, even though it's online.
Okay, thank you. That's good to hear. And with this, I'd like to close this academic ceremony.
Thank you.
Thank you.